Welcome back to the third and last in my series of videos on psychology in the Hunger Games, as we count down to the premiere of the fourth and last of the Hunger Games movies. Today we will look at the question of how real the prospect of true hijacking is, and see what steps neuroscientists have taken towards being able to implant or modify memories.
Part 3. This is our topic. Is hijacking possible today? So, is it possible that in the near future unethical scientists like those of the Capitol could do to humans what was done to PETA? Is it possible to rewrite someone's memories in such a specific and sinister way? How close are we to hijacking technology? Well, in a way, we've already establishing that hijacking is a form of fear conditioning, and that fear conditioning is an important component of post-traumatic stress disorder. So in one sense, we can say, yes, we can do this. After all, Watson and Rayner showed that back in the Little Robert study in 1920, that fear conditioning works in humans, and PTSD is diagnosed in many people annually. But what about the more extreme version we see in PETA with the greatly exaggerated symptoms? For example, the complete reversal of his affections. He once loved Katniss enough to die for her. Now he has been programmed to fear and hate her, so that he tries to kill her on sight. What about the completely false memories he has of her being his enemy and killing his family? Well, happily, that so far remains in the world of science fiction. That degree of behavior and memory modifications just isn't possible. We do not have a tool like Tracker Jacker Venom that allows such manipulation in humans, at least not yet. But there have been some recent studies in mice that have apparently created, or in some cases rewritten, memories by stimulating brain cells. Now, not with a chemical like Tracker Jacker Venom, but by going directly into the brain and stimulating individual neurons with either electricity or light. And so I'm going to tell you about two such studies. The first was by De Lavillon and colleagues in 2015, where he actually created a memory, or what looks like a memory, in sleeping mice. The second was by Redondo et al., published in 2014, where they were able apparently to change good memories to bad memories in mice, and vice versa. So, to understand this first study, you need to understand two parts of the brain. So I'm going to familiarize you with those. The first is the hippocampus, this little turquoise part right here, as shown in the mouse brain or in the rodent brain. And this is a part of the brain known in both humans and lab rodents to be involved in memory, particularly involved in memory for location. Now, the hippocampus has specialized brain cells, specialized neurons that are called place cells that fire when an animal is in a particular location in a test arena. For instance, here is an example of a test arena. So imagine this is a box and you've got a mouse wandering around inside it. So what's happening here is that 4 individual place cells have been tagged or labeled and are designated by a different color, and the scientists are recording from them as the animal walks around. So each dot here represents a particular place cell firing. So as you can see, the green place cells fire mostly when the animal is down here toward the bottom of the box. The yellow cells fire more when it's up here. The blue cells represent this top corner, and the red cells represent this part. So these cells seem to be, the firing of these cells corresponds with the physical location of the animal, and they seem to be an important way that the animal keeps track of its location and forms memories for what happens in particular locations. So place cells are fire differentially depending on where the animal is. Another thing that's very important is that these place cells also fire spontaneously when the animal is asleep, probably when it is dreaming, and that will be important for this next study. The second part of the brain you need to learn about is the medial forebrain bundle, or the MFB, which is this part of the brain here that begins in a spot here called the ventral tegmental area and sends axons up and communicates with this part of the brain here called the nucleus accumbens. So a whole series of neurons in this part of the brain. This pathway is associated with reinforcement or reward. So it is activated when an animal does something enjoyable, eats tasty food, or mates with another animal. It's activated in humans if you're in Vegas and win a jackpot, or if you eat something delicious, particularly when you're hungry. It's also activated by most drugs of abuse, such as cocaine. So it explains the appeal of those types of chemicals. So basically, whenever we do something that activates this medial forebrain bundle, we immediately want to do that again. So here is what the researchers did. They took a mouse, let it wander around in the arena, and identified place neurons that were associated with a particular spot, here indicated by the pink circle. So they found the place cells that fired whenever the animal is in that particular location. Then they took the animal out of the arena, put it in another cage, and let it go to sleep for an hour. While it was asleep, they were recording from those same place cells, identified them, and waited for them to spontaneously fire during sleep. Whenever the cells fired, the scientists would use another electrode to stimulate this medial forebrain bundle right here, so that the two groups of cells would fire together. Now, there's a saying in the world of neuroscience: cells that fire together wire together. So their hope was that this co-activation of these two sets of neurons would form a type of association between the place cells and this reinforcement area. So then they wake the animal up, put it back in the arena, and this time you can see its behavior is different. It doesn't wander all over the place. It walks around, but when it goes into the pink zone, it stays there. It is behaving now as if it has a memory of something very pleasant happening there. Even, of course, even though it has no such memory, it just has what the scientists did when it was asleep. So it's almost like a false memory has been implanted. Now, the next experiment is even closer to PETA's hijacking. Redondo et al. Were also working with mice. Actually, they were specifically working with male mice. If you want to give a male mouse a pleasant memory, you give him a sexually receptive female mouse. So that's what they did. The mice in the studies actually spent 2 hours in their home cage with a female. So during these 2 hours, when the mouse is interacting with the female, The researchers are recording from the brain and identifying hippocampal cells that were firing during this encounter with the female. And what they did was they tagged those cells with a special gene that, when it is expressed, allows the researchers to go back and reactivate those same cells later by shining a light into the animal's brain on this fiber optic cable right here. So they identified the hippocampal cells that fired during the sexual encounter. They tagged them so they could be reactivated later. This technique is called optogenetics, and it's a very new technology. So 2 days later, they put the mouse in a preference chamber, and they picked part of it that they designated that would be, when the mouse entered it, those cells would be turned on. They would shine a light into the animal's brain. Now, of course, the light was not hanging there on a light bulb in the chamber. It's going on that fiber optic cable into the animal's brain. I'm just representing it here to indicate the location. So, what happens? Well, initially, the animal sort of wanders around the chamber at will, but once it crosses into that area and the light turns on in its brain, it seems to like it, and it winds up actually spending most of its time there. So getting those neurons that were associated with the female turned back on or reactivated seems to be reinforcing, as you might expect. So now they're going to take that same mouse and do a procedure very much like the counter conditioning procedure we talked about in the last video. So what they did is they took the mouse, put it into a chamber for 8 minutes. And over that 8 minutes, they delivered 4 modestly noxious shocks to the animal's foot. Again, enough to startle it, not enough to cause any injury or permanent damage, but enough to be aversive. So when the shocks were administered, they also turned the light on to reactivate those cells. And so they did this 4 times over 8 minutes. So now those cells that were associated with the female have now been reactivated in the presence of shock. 2 days later, they go back to the preference chamber, same thing. So the mouse goes into the chamber, and let's see what it does. Well, as you can see, its behavior is pretty much the opposite of what it was before. It can wander out anywhere it wants to go, but when it goes into that area and the light goes on, it goes right back out again and chooses to stay most of the time outside of that area. So it seems as if the good memory associated with the female encounter has now perhaps become a bad or a frightening memory for the mouse. And now its behavior is changed. It's behaving as if turning on those neurons is no longer reinforcing. It's something the animal would prefer to avoid. So, is this mouse truly hijacked? In the sense, does it have memories of its girlfriend changed into bad memories, the way PETA's memory of Katniss were changed into bad memories? So, to answer that, of course, we'd have to know whether this mouse is actually remembering the female threatening him or hurting in some way, just as PETA remembered Katniss killing his family. Well, of course, we don't know if that's genuinely what the mouse is remembering because we can't actually ask the mouse that. We can only see where the mouse goes in the test chamber and see if he seeks out or avoids the light that activates those cells that originally fired when he was with his girlfriend. Now, the answer probably is not that the animal is actually remembering the mouse hurting him over. More likely, it's the place-associated aspects of the memory. Remember, these hippocampal cells are associated particularly with memories for places. So, as Takuchi and Morris wrote about this particular study in the journal Nature, light is being used to selectively activate the representation of the where component of a memory and then change its what component. So this is a bit different from Peter's experience, where he presumably wasn't so much the where that was being reactivated. He was actually consciously having memories of Katniss doing horrible things. But still, it is impressive to me that such a brief activation of neurons at the right time can change the emotional component of a memory so efficiently and then produce such drastic behavioral changes in these mice. So, I should emphasize that at this point, no one is trying these sorts of experiments on humans. In fact, these techniques, particularly things like that optic genetic labeling, are not possible in humans. Still, it raises some interesting questions. Manipulating memories is an ethically charged issue. Now, I should say Redondo's study worked in reverse as well. They could change the aversive memories of shock into pleasant memories by if you take the cells activated when the animal was in the shock chamber, reactivate them during the romantic encounter, and now you can get the mouse to want to go into the area where the light is turned on. So turning bad memories into good ones. This could actually be helpful in some circumstances, particularly for a person with PTSD or phobias, anxiety, other fear disorders. So that is one potential application of this if this technique could be adapted for humans. However, there's a darker side to this too. A commentator on the first study we talked about said this: the simple yet powerful experiments by De La Vallione and colleagues not only represent a neat addition to our understanding of memory and navigation, but also pave a path for fascinating, though potentially sinister, techniques of neurobiological manipulation in the future. So, let's take all of this together now. Let's take all of the psychology we've talked about over the series of 3 videos. What ultimately can we learn by looking into the psychology of the Hunger Games series? Well, the Hunger Games, of course, is dystopian literature. And part of what dystopian literature is supposed to make us think about are the potentially sinister applications of modern technology. So I don't think we have to limit ourselves to the more obvious topics of the Hunger Games, things like the powers of the government, individual freedom, the influence of the media, the power of propaganda. We can think about what our scientific advances mean and what ethical questions they raise, and particularly when it comes to the areas of memory studies. Second, I think understanding hijacking helps us understand the power of fear and what it can do to people. We can understand more about how post-traumatic stress disorder develops and the long-term damage that exposure to violence can do. We can also learn something about what helps treat it. Third, when we connect Katniss's and Peeta's story to both the history of psychology and to modern research in the field, like behavioral neuroscience, that can enhance our appreciation of Katniss and Peeta's story. We really love these characters in this very popular series. We appreciate them more when we know a bit more about what makes them tick and how it relates to modern psychology. And in turn, the Hunger Games trilogy can also inform our understanding of psychology. If you're interested in these types of topics, you can look at these examples and they form something you can relate to, form something that helps you understand more about conditioning, trauma, memory manipulation, in this case, even things like neuroscience. So, this brings us to the end of our video series, and I hope you have enjoyed it. So once again, I will remind you of the resources you have if you want to know more. We have the book, Critical Insights in the Hunger Games, coming out next summer. We have more videos coming out on psychology and popular young adult literature. If you would like to be added to the mailing list to get alerts for those, please send an email to divergentinDepth@gmail.com. So this is the email to be added to my psychology and young adult literature email list. So thank you very much, and hope you enjoyed it.
All right, thank you for watching, and please contact me for comments and questions. I'm Dr. Louise Freeman, Psychology Department, Mary Baldwin College, and for the last time, may the odds be ever in your favor.